Okay, so welcome everybody. And thanks to Dan for the lovely invitation. It's really great to be back on campus. I knew it would be a good day when the first person I met this morning was Jeremy Thorner, who I haven't seen in almost 10 years. It's great to be back and look forward to discussing with you some of our work that really speaks perhaps quite nicely going from the kind of cellular level that we just heard, single cell level, to much more broadly to human populations, and how in humans and across, of course, organism, there is a profound effect of early life experiences that might affect our behavior and our health across the lifespan. And so, of course, preaching to the choir, showing you this kind of picture, but this is obviously the sort of the developing brain from a newborn to over the first few years of life. I have a 3-year-old, so I can tell you that there's lots of stuff going on that makes it challenging to deal with. As you probably all know, and then with a much more sophisticated and actually quite trimmed down version in the adult brain. And so just from this very simple slide, it's clear that these early years are years of wonder, that these early years provide a lot of opportunity to do things right or to do things wrong, whichever way you attitude is. So these are just some numbers that really in these first years of life, there are more than 700 neurons that neural connections per second, and by year 3, the child's brain is twice as active as the adult brain, maybe even more if you're sort of slow thinker like I am. And of course, then that makes it clear that these early life experiences that really determine the capacity of the brain. And there's lots of work that speaks to this. Perhaps one of the more intriguing one that motivates some of our work is that the effect of early life environments that relate to early life socioeconomic status and poverty. And this is data from a very small study, but nevertheless quite instructive. That if you look, there's a clear stratification in the cumulative vocabulary that these children speak, depending on whether they belong to low SES families, working class families, or then children of professionals, so high SES families. And you can see that this gap is only getting wider as the children age. And so epigenetics has emerged as one of the key mechanisms that people have been starting about thinking about about mechanistically how these early life experiences can have such a profound impact onto our life behavior and health across the lifespan. So what I'd like to do today is to talk to you about epigenetics, why epigenetics is emerging as a key factor between the mediator in the interface of genome environment. Show you some data about epigenetic variation in adults and how some of this might indeed be traced back to biological embedding of early life experiences. Then talk about perhaps the biggest challenge or opportunity, depending which way you look at it, that we face if you kind of do the work that we do, which is epigenetics on a population level, which is trying to understand what can we learn from the periphery about the brain. And then move on to share with you some really emerging data about epigenetic aging and how this might relate to some of these early life experiences. And finally, if we have time, give you a glimpse of what I think is another really exciting area that we're engaged in that speaks to the fact that the genome and the epigenome might actually be in many cases much closer than people think. So the definition of epigenetics, of course, for those of you who have followed the field, is quite controversial. This is one that a lot of people like. As you probably already heard, I'm German, and I grew up in Germany, and by virtue of that, I should love definitions. I actually hate definitions, which is probably why I'm in North America. But for those of you and so what we're really interested in is then the social aspect of this, and basically how can life experiences influence the way our genes are expressed? How do experiences get under our skin? And so what we're really trying to understand is how an experience affects the biological pathway all the way down to the gene expression, but of course, perhaps even more excitingly, how this in turn then goes back to affect biological pathway and the kind of experiences that we are exposed to or that we basically seek out. And so, as you probably know, there are many, many different aspects of the chromatin, of the packaging of the DNA that contribute to make up the epigenome. Those are shown here. We heard already about some of them earlier today. The one that I want to focus on today is DNA methylation, and for the purpose of this talk, focus on DNA methylation primarily or exclusively in the context of CpG methylation. And so if you look at a textbook, this is perhaps the best understood epigenetic modification, presumably in particular as it relates to gene expression. If you want to think of a gene as a light bulb, if the gene is on, it makes lots of mRNA. There's very little DNA methylation. If you want to think of the DNA methylation as the regulator of this dimmer here, if you get some methylation in the gene promoter, it's pushing the dimmer down, and the light bulb gets dimmer and dimmer until it's all the way off. So this is sort of the canonical model. Lots of promoter methylation leads to a reduction in gene expression and vice versa. In the context of social epigenetics, it's important to keep in mind that if we look at two different cell types, and these are just two of the ones that we're going to talk about today, so a blood cell and a white blood cell and a neuron. Of course, the gene expression patterns are very different as they are being established over the course of development. And many of those gene expression patterns are basically maintained by different epigenetic modifications. So when we talk about the environment affecting the epigenome, we're not talking about the environment changing a neuron to a white blood cell. Much rather, we talk about the environment perhaps changing the intensity of this light bulb here, in this case in both cell types, or in another case it can be only in one cell type going upwards or downwards. So this is what I want you to keep in mind as we talk about this. Sometimes when you sort of read the more popular mainstream literature about epigenetics, that's actually not being made very clear. So there are many, many different environments that have been shown to affect the epigenome to various degrees. First of all, there is really striking data both from animal models initially, but more recently from humans, that the nutrition and the lifestyle of the mother during pregnancy can affect the epigenome of the offspring. A number of the papers that have come out have shown that people that were born during the Dutch famine at the end of World War II to mothers that basically had very little nutrient uptake had epigenetic changes all the way up to age 60 in their white blood cells. Similar data are numerous studies showing that maternal smoking can actually change DNA methylation in kids and their offspring. Maternal smoking during pregnancy can change DNA methylation in their kids, and again in quite lasting fashion. Perhaps the single most exciting aspect that brought this field of social epigenetics into the mainstream is the work of Michael Meany and others that showed that the maternal care, the quality of the maternal care, can affect the epigenome of the offspring. This, of course, you're probably familiar with the very famous licking and grooming study in rats, published in 2004, but similar aspects are have been found in humans, and I'm going to show you a tidbit of our data that speaks to that. Furthermore, we heard about identical twins earlier today, and discordance and concordance in phenotypes. It turns actually out, work by Manuel Estella and Art Petronis, that identical twins, when early in life, have very concordant epigenomes, but as they go through life, their epigenomes become much more discordant, and in particular, in those twins that happen to be separated early in life. So again, suggesting that this early life environment gets under the skin to modify the epigenome and potentially lead to different health and behavioral outcomes. So this then led us to propose the following model, which is sort of the core of what we do in our ecosystem of collaborations and studies, that phenotypic variation really at least is an integrative interaction between, of course, our genetics in our environment, but also our epigenetics. And I'm going to show you the majority of the data today on this axis, some data on this axis, and we're actively in the process of putting all of this together. As I mentioned, this field has become quite interesting for the mainstream public. And for those of you who are interested in epigenetics in an evolutionary context, and I recommend this article from Time magazine. This came out 3 years ago, really speaking about a lot of the epidemiological studies that suggest that epigenetics might be even inherited across generations. So putting our model to the test, it occurred to us some years ago when we started this that it actually wasn't clear that on a population basis there was a lot of variability in the human epigenome. We and others have since shown that indeed individuals do vary in their DNA methylation pattern in the same tissue. We estimate the extent of variability, depending on the cutoff, to be between 5 and 8% of all CpGs that we look at. Perhaps not surprisingly, this variability and to some extent can be tissue specific, and we're going to come back to this in a bit. Having established, of course, that there is epigenetic variation in the human population, we then ask the next simple question. Well, to what extent is this textbook model that I showed you about more methylation causing less gene expression actually true when we look on a population basis? And when I say population, we're talking about between 100 and 300 people in the majority of our studies. And what we found is that it depends through which lens you look at this. So first of all, if we look at all 25,000 genes and their associated DNA methylation status in a data set of 100 people and their white blood cells, we find that there is indeed the expected overall negative correlation between methylation and expression, shown here. And this is one example where you can see that this gene expression goes down as it's becoming more methylated. When we then ask a slightly different question, well, what about if we look at the correlation for one gene across and the DNA methylation for an expression across all 100 people? We see quite a different picture. So first of all, we see that there are indeed some negative correlations, and the different colors just indicate various levels of FDR correction. But to our surprise, we also found some positive correlation. In other words, where on this individual gene level, more methylation would be going hand in hand with more expression. When we look closer at the literature, that wasn't perhaps so surprising because there was an emerging appreciation for this to be happening. Perhaps more surprisingly, we also found that there was a large number of variable CpGs. So on the y-axis here you see standard deviation. There was a large number of CpGs whose DNA methylation status really wasn't correlated with the expression state of its underlying gene that it's supposed to regulate. And so, again, this was very surprising when we saw this, but since has been replicated by numerous other studies by other investigators. And so then the question arises, well, why is that? Why is, when we look at the population level, that this seemingly so straightforward relationship isn't really there? So we, of course, saw that, well, the simplest explanation is that we really need to look at many of these other modifications in addition to DNA methylation to perhaps get the correct picture. That might well be true, but I think there's more exciting and not mutually exclusive possibilities as well that really raise the question, what really are the origin and the consequences of variable DNA methylation? And drawing back on some of the model organism work that has been out there for a while in fruit flies and even in yeast, we then hypothesized that perhaps the reason why there is this apparent disjoint is that what this DNA methylation is it's not only recalling the history, as I will show you throughout this talk, of what happened to us, but perhaps it might also prime us for a future response. And we're in a position to actually test this rigorously because we had actually used those same PBMCs from which we got the DNA to measure DNA methylation and the RNA to measure gene expression. We had stimulated them with various agents that engaged the TLR pathways, which you probably know is a core component of the innate immune system. And so what we published 2 years ago in PNAS then is the finding that if we look at this DNA methylation in these PBMCs and we compare their IL-6 production in response to this TLR pathway engagement, we find a highly correlated relationship as indicated by these very skewed p-value distributions for both TLR9 agonists and TLR4 agonists. These survive rigorous multiple testing and involve functional pathways involved in the immune response. So this was very exciting because it really suggested that perhaps what this DNA methylation indeed is doing, it's priming our cells for the event, in this case, the stimulation of the innate pathway. What we're now doing is to measure before and after the DNA methylation as well as the mRNA expression, so stay tuned for this. Hopefully, we'll be finished very soon. In this same cohort, we also found other factors, more lifestyle and demographic factors that were associated with DNA methylation. So things like gender, ethnicity, perhaps not entirely surprisingly. Gender, I should mention, we of course take out the sex chromosomes, so there are sites on the autosomes whose methylation status is associated whether you're male or female. Perhaps more excitingly for the kind of framework that our work is positioned in, we also found that things like perceived stress and total cortisol during the day were associated with DNA methylation in these white blood cells, as indicated again by these highly skewed p-value distributions. And again, we were able to identify specific CpGs that survived multiple testing correction. The reason why we're really excited about this is, as I mentioned initially, about these psychosocial factors, is that the way we positioned our work is really to understand how things like early life socioeconomic status, so whether you grew up in this kind of dwelling or in this kind of dwelling, or early life paternal stress, whether you grew up in this nice nuclear family with a dog, or a family where the parents constantly yell at each other, whether those indeed, as one might predict, alter the DNA methylation pattern to ultimately perhaps identify affect childhood temperament, behavior, and trajectories. And so over the last few years, we've really developed a large body of work and wonderful collaborations, and I'm really standing here on the shoulder of some giants that we were lucky enough to collaborate with. People like Craig Miller and Edith Chen, who used to be at UBC and now are at Northwestern. Tom Boyce, who I think was at Berkeley and then moved to UBC and then is now back at UCSF. The late Clyde Hertzman, who in many ways was a grandfather of this field of genomic embedding, and also Marilyn Essex at the University of Wisconsin. And so collectively, this really allowed us to ask this question: how do the diverse set of early life experiences get under the skin? In my first slide, I did briefly allude to the effect of socioeconomic status on vocabulary. There are actually numerous studies out there that really speak to the fact that early life socioeconomic status has lasting effects on human health. This is one of my favorite ones because just on the pure numbers. So this is a study that was published in 2006. Where researchers looked at more than 1,000 wild medical school graduates from the Johns Hopkins University. And I'm sure you're all familiar with the fact that when you graduate from Johns Hopkins Medical School, you're probably well established in the community, you're making good living, and you're generally a productive member of society. But what was documented is there's a stratification in the cumulative risk of coronary heart disease that you can be seen here in those two lines that really can be drawn back, can be correlated back to whether these people grew up in low or high early life SES in the first 5 years of their life. With ultimately, I think the cumulative risk being almost 2.5-fold higher for the physicians that grew up in low early life SES. And so these other studies then basically led to this idea that there is some biological mechanism, i.e. epigenetics potentially, that leads to this biological embedding of these early life experiences all the way into adulthood. And with this set of studies that we're engaged in, we wanted to put this to the test and designed specific cohort strategies to try to test this hypothesis. And so the first study that we did was with Craig Miller and Edith Chen, where basically, and again, this is a cohort of 100 people, healthy people in the Vancouver area, but there were basically two groups. So there were a group of people that grew up in low early life SES and a group of people that grew up in high early life SES. These people, when we tested them, were all in their 30s or 40s, and we mixed them for their adult SES so that we were able to directly deduce any findings and link them back to their early life SES. What we then did, we measured in white blood cells mRNA expression and asked the question, do we see a difference in the mRNA expression pattern between these two groups? And indeed, we do see a difference. And it's not a random difference, but instead it's a difference where genes that have binding sites for specific transcription factors involved in the immune response are either over or underrepresented. So for example, this set of genes is underrepresented for glucocorticoid receptor binding sites, and it's overrepresented for NF-kappa B binding sites. If you combine those two, it's becoming statistically highly significant. And for those experts among you, this is the kind of Steve Cole kind of analysis of transcription factor binding events in mRNA data sets. Perhaps more important, so this suggests actually that there's a pro-inflammatory gene expression pattern that these adults carry in their white blood cell that is predictive potentially of a heightened immune response. And indeed, when we then take those white blood cells and we challenge them with various agents, such as that again engage the TLR pathways, we do indeed see that folks that grew up in low early life SES for these two conditions have a heightened immune response of their immune system to these engagements. Furthermore, on the whole body level, we see that folks who grew up in low early life SES have a higher output of cortisol throughout the day. And lastly, in a paper we published in 2012, we also find that their DNA methylation pattern is correlated with early life socioeconomic status, as again indicated by this skewed p-value distribution. Interestingly, the current socioeconomic status does not seem to be correlated with the DNA methylation pattern, at least after we correct for differences in white blood cell types. So collectively, these data then suggest that indeed, whether you grow up in this kind of dwelling or in this kind of dwelling leaves a biological vestige in that we see differences in DNA methylation, we see differences in gene expression, and consistent overreaction of the white blood cells of people who grew up in low early life SES, as well as heightened cortisol output throughout the day. Now, when we first did this work, I was fairly new. As many of you know, from Jasper's lab, I came out of yeast biology, yeast genetics, and I got a phone call from a newspaper in Vancouver, and they basically asked me, and this is a newspaper that is read by working class folks, they asked me whether their readers were doomed. And I said no, but anyhow, I had a nice chat with the journalist. The end result was that I was quoted that their readers are doomed, which is not what I said. So this then led me to do two things. First of all, I enrolled in media training. Secondly, we then asked the question, well, is there anything in these people of low early life SES that perhaps provides hope, that provides perhaps opportunities for interventions? And so we then analyzed additional factors in that group that perhaps might reverse some of these findings that we had reported previously. And any wild guesses whether we found something? We did, otherwise I wouldn't tell you. What might it be? That's right, maternal care. So maternal warmth, basically, when these people were exposed to high levels of maternal warmth, their immune response went further down, and to a large extent, their gene expression patterns were also changed back to a level that was more reminiscent of the folks who grew up in high early life SES. And so basically what I always tell people, even if you happen to grow up in these kind of conditions, but you get lots of love from your mom, you're going to be okay. And so I advise people to choose their parents wisely. Easy. So then, of course, the problem with this study is that it's all based on recollection of people. And we did interview the parents very rigorously, but being a new parent, I think nobody would admit that they were not the greatest parent in the world. But then good things happen when you talk to people. So we came across, we met Marilyn Essex, whose body of life work was to set up this Wisconsin study of families and work, which is a longitudinal birth cohort where she followed more than 500 families from before birth, and these kids are now 18 years old. What Marilyn had done when these kids were 16, she had taken some DNA with the idea of doing GWAS studies, but then she met us and she realized that, oh, maybe it's kind of cooler to do epigenetics. And so we started a collaboration with Marilyn, basically asking the question, well, can we see anything in these 16-year-old kids that would be DNA methylation partners that would be associated with the early life environments? Of course, the first question we wanted to ask is early life socioeconomic status. Unfortunately, fortunately for these people, they're all from middle-class suburban Wisconsin areas. There was almost no gradient in socioeconomic status. But there was quite a gradient in this kind of stress that the parents were reporting and that was ascertained by the research assistants throughout the study. And so then we asked the question, can we identify signatures that are associated with either the mom's or the dad's stress in these kids at different developmental stages? And indeed we could. This was published in Child Development a couple of years ago. We could find a signature of the maternal stress during infancy. And these are, in the entire group, we found 139 methylation sites. Again, for the experts among you, this is multiple testing control at below 5% or what we call medium confidence sites between 5 and 20%. When we divided the group into boys and girls, we lost statistical power and didn't really identify too many sites. There was almost no association of the dad's stress with the methylation in infancy. And I gave a talk in Munich at the Max Planck for psychiatry just before Christmas, and somebody asked me, and I never thought about this, could this just be a reflection of society that at the time moms stayed at home? What would happen if you do it now in families where the dad stays at home? Just leave that with you. In contrast, in preschool, we saw quite a different picture. Now we didn't really identify a signature of maternal stress, but instead we found a signature of paternal stress. And while we saw that in the full group, it was much more prevalent and driven really by the girls only. And so when we saw this, is really quite satisfying because as anybody who's involved in child development would tell you, these relationships very much mirror on the epigenetic basis, if you wish, what people in child development know about the interaction between parents at the different stages between infancy and preschool, and particularly as it relates to either the girls or the boys responding or interacting with their mom or their dad. So it was a very satisfying experience in that we could say that there's an epigenetic mirror of what's well known in the field of child development. We are now in the middle of analyzing the next logical step then, well, is there anything of the signature that we can identify in the children that is associated with their behavior, either at preschool, kindergarten, or grade 1? In other words, is there an epigenetic signature that's associated with this child being retrieved and shy, or this girl here being very outgoing and happy? And these are ongoing investigations that we're just writing up for publications. And for this, we turn to principal component analysis to simply reduce the space of queries that we're doing. First, focusing on all the variables that we have for these kids, which are almost 500. And we identified a set of PCAs, as you might expect. And the second of these PCAs was actually driven by anger, negativity, and internalizing behavior. And this particular PCA was coordinated with DNA methylation in these kids, as shown by this skewed p-value distribution. After rigorous multiple testing control, we ended up with one particular gene where there are a number of multiple methylation sites in the 3' CpG island of this DLX5 gene, which is a homeobox gene involved in multiple aspects of development, where we can see increasing methylation, as shown on this heat map here, for these individual CpGs, going from the low anger, high net activity, high internalizing kids to the high anger, low negativity, and low internalizing kids. We have then had the opportunity to test whether there is a developmental replication, because Marilyn Essex actually had taken DNA 3 years later from the same kids. And when we tested this directly, we could see that these relationships actually were also seen in the 19-year-old kids. So we're very excited about this because it seems to be holding up in 2 independent time points in these children. Now, of course, the big problem with when you study epigenetics on behavior is that we really only have access to say, white blood cells or cheek swabs, because nobody wants to give me a piece of their brain. And so similar to the previous speaker and similar to what Matt Stay told us this morning, we then start to ask the question, what can we really learn from measuring DNA methylation patterns in the periphery, i.e. in the blood or in cheek cells, about DNA methylation pattern in the brain? And if you look at the literature, there is an exploding number of studies, even in fairly high-profile journals, that report on DNA methylation patterns from blood, for example, as it relates to neurobiological processes. And so we teamed up with Gustavo Turecki and Michael Meany from McGill University. Gustavo is the director of the Quebec Suicide Brain Bank. But similar to the previous speaker, we decided to first go for the controls, i.e. postmortem brains of people who died of motor vehicle accidents, seems to be the common theme. And for some of those, for about 20 of those, Gustavo also had collected blood cells. And so that then allowed us to basically ask the question, in matched samples, what can we learn from looking at white blood cells? And we focused on these three brain areas, the parietal cortex, frontal, and temporal cortex, to start these studies. So what can we really learn? And what are the major drivers of variability in these kind of data sets? So this is currently submitted. What we turned again to is principal component analysis. So here you see basically the first three principal components and the loadings in the three different brain regions and then the blood. And here you see the sample traits. And so it's very obvious that this first principal component, which contributes roughly 75% of the entire variance in the data set, is basically just depending on whether you're brain or blood, okay? Suggesting that there are, in fact, quite massive differences in DNA methylation between those two tissues, perhaps not surprisingly. The second principal component, you can see that there's a nice pattern in brain, but it's flat in blood. It actually turns out if we bioinformatically reconstruct the ratio of neuron to glia in those brain samples, it's almost a perfect correlation with this principal component, which contributes roughly 8% of the variance. And similarly, if on the third principal component, you can see that they're flat in brain, but a nice pattern in blood, it actually has to do with individual blood differences in terms of the amount of granulocytes that each individual has. So basically what this slide tells you is if you look at the kind of data sets comparing white blood cells to brain regions, the majority of DNA methylation variation is driven by either which tissue you're looking at, and then the next, but quite significantly less variation, cellular heterogeneity in the tissue, whether it's blood or brain. Collectively, I think this is roughly like 80 to 85% of the overall variance. So then the question arises, well, how much room is there for other variables? And so for these people, because they died of motor vehicle accidents, there wasn't really the kind of analysis that we would like in terms of their socioeconomic status and such. And the group is probably too small as well. But what we knew about these people, the one biological variable that we knew about was age. And so it turns out that actually PC4 and PC5 both are related to age, correlated with age. And interestingly, PC4 is correlated to age only in brain. You can see that here. This is increasing age going for each of those tissues. And PC5 is correlated to age in brain and in blood. So when we saw this, it was kind of surprising because really there was not much evidence that there would be different signatures of aging in the DNA methylome in the epigenome. And we were, of course, worried because we only looked at 20 people that this might have been an artifact of using PCA and the kind of correlations we did. But again, good things sometimes happen. And Trey Idegar had just published a paper in Molecular Cell where he looked at white blood cell methylation in more than 650 people as it relates to aging. And so we took advantage of this data set and asked, can we replicate our data sets, our principal components in that data set that was independently ascertained? And indeed, you can see here, the key figure is here, that if we project our eigenvectors onto this data set, we get a very strong correlation with a p-value of 10 to the minus 50 in this independent data set for the principal component that is common between brain and blood we get no correlation for the principal component that's common, that we only found in brain. So this suggests that there are indeed two distinct signatures of aging, epigenetic signatures of aging in the human brain, one of which can be also seen in the blood and can be replicated in a much larger independent cohort. I want to show you then, going away from that highly complex PCR analysis, and show you a couple of sort of more concrete examples of what this really looks like. So this is a heat map that shows, I think, 30 or 35 different CpGs that kind of cover the glucocorticoid receptor gene, which is the holy grail for everybody doing behavioral epigenetics, based on the work of Michael Meany. And you can see these are the different CpGs. These are the this is the white blood cells. And these are the 3 different brain regions, and then there are each 20 individuals within those tissues. And without going into too much detail, I think you can see that this is a highly complex picture, in that there are certainly some regions like this where the DNA methylation in brain and blood is quite similar. But then there are regions like this, where you have no methylation in blood, but highly methylated in brain. And it's sort of, you can find all kinds of examples, including variable CpGs that are variable only in one tissue, but not another. And so I think we're only learning to deal with these data because it's much more complicated than mRNA expression data, because we're really looking at almost the individual CpG side. And what we're now trying to find out is to figure out, can we group some of them to really get more broad ideas of the rules that govern whether a CpG is tissue-specific or not. But I think certainly one cannot say simply, oh, CpG methylation of GR is different or similar between brain and blood. It needs to be much more nuanced. And when we look at this across the entire data set, what we generally find is that there actually blood and brain DNA methylation are not all that tightly correlated. This is a distribution on the level of individual CpGs. You can see they tend to be negatively correlated, but all the correlations are fairly low, although there are some where, of course, they're going plus minus 0.8, and you can see on the minus 0.8, these are discordant CpGs where clearly you get almost opposite methylation patterns whether you look in brain or blood. And on the concordant CpGs, there are some CpGs whose methylation pattern in blood clearly mirrors that in brain. So as I said, I wanted to show this to you because we're only at the beginning to analyze, and I'd love to talk to folks that are familiar with this kind of analysis, what we should do next. If we kind of look more at the entire, so I should say that these are 500,000 CpGs that we measure in these experiments. This is sort of a modified volcano plot, if you wish, where we have the correlation again here, negative and positive correlation, and then variability, because of course for the questions that we're interested in, it's really the variability as opposed to the tissue-specific nature that really is most relevant for what we're looking at. You can see it's actually a fairly small number. We think roughly 10,000 out of those 500,000 where we have both either negative or a positive correlation and an appreciable amount of variability. So again, these are early days, but I think collectively what this suggests is at this point that there's a relatively small number of individual CpGs from blood that report correctly on what we can expect in the brain. And so whenever you see a paper in any of the journals that talks about DNA methylation in the blood as it relates to behavior, neurobiological processes, you might just want to take it with a grain of salt. In fact, Bruce McEwen actually emailed me the other day and wanted us to publish this ASAP because he said in the study section that's one of the biggest questions that they're encountering. So then, having realized that perhaps this simplistic way of trying to sort of use peripheral measures to understand things going on in more centrally might be quite challenging, we're now turning or including an additional measure in our ascertainment of the epigenetic biology, and that is the idea that was put forward by Steve Horvath roughly a year and a half ago in a really wonderful genome biology paper, where Steve has proposed that there is an epigenetic clock that ticks in all of us. And that epigenetic clock, by virtue of the way he designed it, is basically tissue independent. And so you don't have to worry about, is it in the brain or is it in the blood, if you think about it. And so Steve has really pioneered this, and now many people are jumping on this bandwagon trying to understand what is the nature of this epigenetic clock. We just submitted basically a paper where we bring in our thoughts and in the context of what's known in the literature. So basically, what underlies any age-related epigenetic change is, of course, a DNA methylation mark that in this particular case, for example, increases with age in the population. There has been this idea of epigenetic drift that has been around for a while. Basically, it postulates that monozygotic twins actually start becoming more divergent in their DNA methylation patterns, as I told you earlier. And so that certainly could underlie some of these phenomena. But of course, as you will appreciate, this kind of site, even though in the population might go up, it's not really good as an indicator of the epigenetic clock. The kind of sites that Steve Horvath identified and that really make up the epigenetic clocks are shown here. So basically, these sites in everybody more or less behave the same and increase or decrease with age. And then you can think about this basically in the context of chronological age. You would then predict that the epigenetic clock, by virtue of the way it was designed, is quite concordant with the chronological age. But then you can also speculate that there might be some folks who are actually epigenetically young, so for example, Jeremy, and epigenetically old, and that might indeed go hand in hand with their health status. So the epigenetically young people might actually have a better relative health, if you wish, than the average population, and the epigenetically old people might have a worse epigenetic age. And Steve Horvath has since shown that people with high BMI actually tend to be epigenetically older. Interestingly, people that with PTSD tend to be epigenetically younger, which I think is very curious. Now, we then used a cohort of 400 people that we have working on together with Greg and Edith and tested their epigenetic age. These are monocytes, so highly purified white blood cells. You can see that indeed there's quite a very good correlation between their chronological age and their epigenetic age, as ascertained by Steve Horvath. Steve has this wonderful, is very generous with the community. He has this all available on his website, so it's very easy to do these kind of measures. There is a bit of a skew, and we're talking to Steve about this. He thinks it might be because his clock is equilibrated for all tissues, and we have a very specific isolated white blood cell tissue. But I think regardless, you can appreciate that there are certainly individuals whose epigenetic age is varying quite a bit from their chronological age, both being epigenetically much older or epigenetically much younger. We then basically, again, at the beginning of this, we ask, well, is that in any way, shape, or form related to the kind of lifestyle or psychosocial factors that we're interested in? And indeed, what we're finding is that if you look again at early life socioeconomic status, we find that if you grew up, and again, these are people between the 30s and 50s, if you grow up in low early life socioeconomic status, you're epigenetically about 2.5 years older than people who grew up in high socioeconomic status. And while it's early days, just to put that in perspective, there's a paper in Press in Genome Biology from the British group that suggests that an acceleration of 4 years of epigenetic aging translates into 25% increase in morbidity. So that perhaps helps you to appreciate what this could mean ultimately. Again, we don't see much of a difference between current socioeconomic status, whether it's high or low as it relates to this epigenetic clock. Interestingly, when we take advantage of our study design that I showed you earlier in this group, we can see that there's almost like a compound effect of low socioeconomic status, in that the groups that are switching have intermediate accelerations of their epigenetic age, as opposed to the groups that are either always low or always high socioeconomic status. So this just gives you a flavor where we're going now with this, to basically complement the studies that try to understand what we can learn from the periphery about central. And I want to just use the last 2 minutes to talk to share with you some of the data, because they actually originated down at Triple Rock way back when I was a postdoc here. When I met, many of you will remember Hunter Fraser, who was one of the star students in Mike Eisen's lab at the time. And we developed a close friendship and have been working together ever since. But Hunter and I, a few years ago, started to think about, well, how can we bring genetics and epigenetics together? And to do this, we basically took advantage of the HapMap cell lines and measured the DNA methylation patterns in the Caucasian and the Eurubans, I think 60 each. And what we found is that, surprisingly, in these lymphoblastoid cell lines, roughly a third of all CpGs had some kind of population difference, which then would suggest that perhaps to some extent this might be driven by underlying genetic variation, because, of course, these cell lines had been out of the human body for many, many years, generations. And so in a paper that we published in Genome Biology 2 years ago, what we found is that indeed there is heritability, in a sense of narrow sense heritability in the epigenome. You can see this here. This is the Eurubans. These are the Caucasians. That there are roughly 1,000 sites in each of those populations where the correlation between the offspring methylation is greater than you would expect by chance, for the parent-offspring correlation, greater than you would expect by chance. Consistent with this, we then, this is one example where you can see this particular site has very good correlation as it relates to mean parental methylation and the offspring methylation in the Eurubans, but not in the Caucasians. And by and large, what we found is that there's really very little in terms of this heritability that the two populations share. So we conclude from this that there's common heritability of DNA methylation, and we think it affects roughly 8% of all CpG sites and indicates really genetic control of polymorphic methylation. And as I said, this has been since been shown by others as well, and so we feel very confident in this finding. And consistent with this, Hunter then was also able to identify actual allelic variants that are associated with nearby CpG methylation sites. So for example, if you look at this particular site, you can see that this is the methylation status, this is the allelic variation, the allelic status. You can see that in both populations, there is an increase depending on which allele you're associated with. The same site that has no heritability between Eurubans and Caucasians actually shows we identify mSNPs in the Eurubans, but not in the Caucasians. So again, this suggests that some DNA methylation marks are under genetic control to nearby SNPs, but it's not as simple as that because there is an additional genetic background likely to affect this. So now we have another paper with Hunter that we're just about in the last round of revision where we use next generation sequencing to understand these things more broadly, and we have identified roughly 20,000 new of these mSNPs. So very excited about this. But just to summarize what I've discussed with you today, I hope that you got a flavor for why so many people think that epigenetics is an integral component of human health and disease. We talked about epigenetic variation in adults and how some of this might be tissue specific and is associated with many different variables, including lifestyle and psychosocial factors. We touched upon that contrary to the textbook, DNA methylation and gene expression are not tightly linked across populations. I think that's very exciting because it gives enough work for both of those of us who studied mRNA versus those of us who studied DNA methylation. Makes it more exciting. How some of this variation might be due to genomic embedding of early life experiences such as early life poverty or early life parental stress. We talked about the challenges. Reporting from peripheral DNA methylation measures onto central ones, and touched upon the epigenetic clock and the idea that there might be discordance and concordance between the chronological and the epigenetic age, and how some of this concordance might be driven by early life poverty again. And lastly, quickly touched upon how genetics and epigenetics in many cases are much more tightly linked than one sort of. So I just want to leave you with a bunch of issues to consider. And we can talk about them later, but the one I want to emphasize very clearly here that everything I've shown you so far is correlation. There's no implication of causation, and we're starting to think about how we can use more sophisticated study designs to really get to a more causal, understand a more causal, whether there's a more causal relationship. And I just want to finish with the acknowledgements. I hope I appreciated all the collaborators along the way, but here are the people in my lab who did all the work, and these are the collaborators whose work I talked about here, and funding agencies that allowed us to do all of this. Thanks a lot.
Thanks, Mike, for a very far-ranging talk. I'll start with Jeremy, the epigenetically young guy.
Now I'm in trouble.
Now, retribution. No. I have a very specific question. There are only 2 methyl donors in cells, S-adenosylmethionine and tetrahydrofolic acid. So my question is specifically about whether or not either the level of transcript or specific alleles of the enzymes that produce those 2 donors of methyl. Is there any correlation between the degree of methylation you've seen in the different samples and
Sure.
that particular issue?
So that's a great question. In fact, early data would suggest that there is a relationship. And those early data came mostly from cancer studies. In our population, unfortunately, because they're mostly designed by psychologists and psychiatrists, nobody measured, for example, the blood level of those precursors or the compounds. Certainly, there's data out there about MTHFR, the famous MTHFR genotype, I forgot which one it is, being associated with changes globally in DNA methylation. We have never seen that, but then we realized that especially in Canada, of course, everything is folate fortified, like the flour and everything. And so we suspect that it might be that there might be an effect, but only when you go at sort of threshold levels of folate. And so we're actively thinking about populations where perhaps there isn't that kind of saturation in the blood. Does that answer the question?
Hi there. So have you looked at methylation and if a child was breastfed versus formula fed? From what I understand, I feel like I've heard that poverty is associated with a higher prevalence of formula feeding, and so is that perhaps what you're seeing? And then when you have this like maternal care, maybe those are the children that were breastfed or something like that.
In our cohorts, we really, so the Wisconsin cohort, we wanted to look at this, but at a time, actually, I think this, from my understanding, it was a time of prevalent formula feeding in most places. This was before, all of, in our newer cohorts, we have a new cohort with 3,000 3-year-olds that we're just starting to look at. We have these data. We haven't actually analyzed them yet, but it's a great question. And you might be, of course, perhaps aware that Nestle, of all things, just gave $150 million to epigenetic research and infant nutrition. So take it as what it's worth.
Mike.
So, as you know, there are species that have the extreme phenotypic plasticity as adults. Ants that switch castes and fish that switch sexes and things like that. That seem to be associated with major shifts in sort of the epigenetic landscape in the brain. Is there any thinking about why, whether humans are missing something that prevents the kind of rewiring of your brain in that way as an adult, or if we've failed to tap into the appropriate capacity to do that or something?
Yeah, well, we're missing royal jelly, which, as you know, there was a great paper in Science that talked about royal jelly actually inducing changes in DNA methyltransferase expression and between the honeybee and the workers. That's a great question. I think, as you know, the field of ant epigenetics has been extremely active. People like Danny Reinberg and all these people getting into it. I think the community is only just starting to think what epigenetics could mean in the context of human evolution and plasticity in human behavior and human brain. So I think there is a lot of work still to be done. It's a great question, though.
There's a lot of interest lately in...
we have two people speaking, sorry.
There's a lot of interest lately in living forever. And like Google Genomics is trying to collect genomic samples of people who have lived very long. But Google Genomics is not looking into the methylation status of the genome. So do you think that they might be better to do so?
Well, I wouldn't, I mean, as you know, Calico, or whatever they're called, they have very, very smart people like David Botstein and others, so I wouldn't want to say that they're wrong to do anything. But we are actually starting to, so we just got funding to look at the epigenetic clock in 30,000 Canadians, for which we also have allelic variation data. Those are normative aging, so it's not like, you know, the supercentenarians and those kind of people, but I think it nevertheless will give us an appreciation to what extent variation in the epigenetic clock is linked to underlying allelic variation, so the ApoB1 and those kind of genes that people talk about, yeah.
Do we have another question? Or was somebody else?
Yeah, this is a very interesting talk, thank you. This may be somewhat tangential, let's say slash random question, but I'm just thinking about this. It's been argued that the human genome or human brain evolution may be, in a sense, buffered by culture, so that culture provides a kind of buffer that allows for increased variation in certain ways and in certain genes. And I'm wondering, as one looks at the role of cultural adaptation or, species that, let's say, change from individual selection to group selection and those kind of things. If there is an alteration in kind of methylation patterns or the extent of heritability of methylation, or if people have looked at that in addition to genomic change.
Right, that's a fantastic question. And I wish I could, I actually do have some resemblance of an answer for you, but I can't really talk about it because my collaborator is horrendously worried that we're getting scooped on this. But we did a study that touches on this by comparing Pygmies and Bantus methylation profile in Africa, and in particular, some of those, so that some of them live in the forest and some in more agricultural areas, and there's a particular group that actually switched, and so that allowed us to decipher what contribution in that evolutionary context of Pygmies and Bantus would come from who you are genetically versus where you live. And I think that starting to touch upon the kind of question that you're asking, with the limitation that, of course, we did it in white blood cells because these were not postmortem brain samples. But I can tell you that the results are very interesting and suggest that there is a contribution of both who you are and where you live.
I have kind of maybe methodical question. How people distinguish between methylated sites and sites which were methylated and then demethylated?
Well, you mean evolution? Like,
no, in an organism, because there are demethylation is going
so
on, right?
there is certainly demethylation, although, that has been controversial for many, many years. Initially, people thought once you're methylated, but it seems to be that there is certainly active demethylation, and numerous people have been doing some very important work on this. We haven't really looked, so the way you would have to do it, of course, is to look longitudinally in the sort of same sample to get a bit of an appreciation for this, which we haven't done. But by and large, so what you in our data, it's all steady state.
But like if you do bisulfite sequencing, basically we cannot distinguish between the sites which were methylated and sites which were demethylated later, right?
No, you would have to do this.
We can distinguish.
Not sure whether. Because there's of course a huge wave of demethylation and remethylation that's occurring early in development, and then there's the complexity of that also being in part hydroxymethylation, which is an intermediate state between the kind of methylation that I showed you and demethylation that's done by the TET enzymes. So I think that's probably what you want to look at to get an idea of the demethylation, if you wish.
Thank you. Thank you.
So you mentioned you saw an allelic bias in the methylation status within the HapMap cell lines, like one allele is more methylated than the other. I'm wondering if you look at your sites which were correlated with, like where the epigenetic status was correlated with environmental Is there an allele which is more preferentially biased to be affected, or are they both affected equally?
Excellent question. We're starting to do this now. Of course, in the HapMap, we had the advantage that they're completely sequenced. So generally, we have much better distance between these fairly low density methylation arrays that we're using, which they interrogate 500,000 CpGs, but that's only still 3% of all CpGs in the human genome. So what we're doing now is, in all our studies, we're using SNP genotyping to ask those kind of questions. And in fact, we have a paper in press that suggests that if you take the BDNF, the brain-derived neurotropic factor, and you combine its allelic status and its methylation status, it better predicts some brain-related process as ascertained by imaging. So that's definitely the next way. That's why I put those three things together.
Okay, we have coffee waiting for us out in the way. Come back at 4:20 to hear the last talk by Daniel Geschwin.